Once upon a time, there was a poor and hungry traveler who came to a village looking for some food and shelter. He said, "I can make a delicious soup from an ordinary stone. I just need some water." The people in the village were skeptic, but they gave him water. He said, "The soup is getting tasty, but it could use a few potatoes. Can you spare some?" And someone gave him a few potatoes. He said, "The soup is getting really tasty, but it could use some carrots. Can you spare some carrots?" And someone gave him a few old carrots. He said, "The soup is getting almost ready now, but it could use some chicken. Can you spare any piece?" And someone gave him some leftover chicken bones. He stirred the pot and said, "Here, the soup is ready. Come and enjoy." This stone soup tale is an old fable common across many countries. You may wonder, what does it have to do with publishing science? If you stick around, I'll make it worth your while. Thank you for inviting me to present at the Karolinska Discovers. It's an honor. I tend to present those as host and a moderator for KI, and I'm really thrilled to share with you the story of our recent publication. If you asked me 2 or 3 years ago if I'll remain in academia and if I'll publish something after my postdoc, I would say it's very unlikely. Today, I want to share the very unlikely outcome of our work of the few years. It was unlikely on several levels. One level was that after the postdoc, I had frankly no resources and no perspective to hire people to run my own lab. And it was also unlikely on the conceptual level, because why would you look at blood vessels in ALS, which is a neurodegenerative disease, right? Most of the time, people are just looking at the neurons which are affected in ALS, because the definition of ALS is that the motor neurons that are connected to muscles, they're located in the cortex and the spinal cord. These neurons are degenerating specifically, which leads to paralysis and eventually death. So you need to have both upper and lower motor neurons affected to fulfill the definition of ALS. And quite right so, people are most of the time studying the neurons. When we're making an enormous progress studying the mechanisms within neurons that lead to the degeneration. So mitochondrial dysfunction, misfold of the proteins within neurons, impaired axonal transport within the neurons. These all contribute to the dying of the neuron, right? And vast majority of people in the field of ALS are looking at the neurons. But this is the central nervous system. And the way that the neuron function depends on support of many other cells, including microglia, oligodendrocytes, and also blood vessels, because they deliver the oxygen and glucose, the energy to the power-hungry neurons that need to function properly. And perhaps if this stressed neuron is not receiving proper blood vessel support, it's likely to exacerbate the disease. But still, very, very few people were looking at blood vessels to answer questions about ALS. And there are some evidence, even in the clinic, where patients with ALS or FTD, which is a related neurodegenerative disease, they display signs of decreased blood flow in the brain, they reduce glucose uptake, sometimes even enlarged perivascular spaces, and immune cell filtration. So this told us, yeah, perhaps there is a vascular component happening in ALS that is highly underappreciated. So we took a step back and tried to see which cells are activated during ALS in which sequence, right? So we took bulk tissue transcriptomes on RNA level from sporadic ALS patients, as well as 2 mouse models. One of them has a motor phenotype and the other has more of a cognitive phenotype. And there we took those bulk tissue data and compared it with cell-specific gene ranks. So from single cell sequencing, you can tell this given cell type, like microglia, has this and this specific markers that differentiate them from other cells. So now you can take the bulk tissue you can look at differentiately expressed genes, right? Rank them and see, okay, in these differentially expressed genes, which cell type is overrepresented? When we looked at the sporadic ALS patients, we saw these typical signatures of reduced excitatory neurons, transcripts, as well as induced microglia and astrocytes, these typical signs of neuroinflammation happening during neuronal death. But when we looked at vascular cells, it turned out that out of these 4 vascular cell types, endothelium, vascular smooth muscles, and pericytes, the perivascular fibroblast cells had the highest cell enrichments. Right? So perhaps there is a vascular component happening in sporadic ALS. But now the question comes, what is the order of activation of these cells? Because now we're just looking at the end stage transcriptome from a deceased patient. So when we looked in the mouse models, we saw that, yes, we have the microglia neuroinflammatory induction, but it happens relatively late during the disease. But what happens before is the induction of vascular cells. So this enrichment of gene expression specific to perivascular fibroblast happens long before you have neuroinflammation, long before you have neurodegeneration events, pretty much at the time that the neuromuscular junctions are decoupling from the muscle. And we were able to confirm this early versus late events looking at another mouse model. So we were pretty confident this could be the case. So now let's have a look at these fibroblasts, because these cells were only recently discovered pretty much through single cell sequencing efforts, and they were just mapped to be localized between the astrocyte enphy that are surrounding blood vessel and the endothelial and the mural cells. When we looked a bit carefully in the spinal cords of mice, we saw that these perivascular fibroblasts, shown in red, are really located between these 2 layers of extracellular matrix called the basement membrane. It's the kind of a scaffold that supports these cells. So their fibroblasts are surrounded by the astrocyte basement membrane layer and the mural cell basement membrane layer. So we have this working model here that we have the astrocyte enphy, the 2 layers of basement membrane, and the fibroblasts are located in between. Since we knew from single cell sequencing which genes are specific to the fibroblast, we could look up those genes individually. And for example, we have a bunch of genes that are specific to perivascular fibroblast. The specificity is shown in the columns. And we could see that these genes are indeed regulated in end-stage sporadic ALS patients, and they have 2 different groups of induction. So one group is induced relatively early, before the onset of symptoms in ALS mice, and the other group is activated in the later stage of disease, more or less the same time as the neuroinflammation response. And we were also able to confirm that these early versus late separation happens in the other mouse model. But these are just computational predictions, right? We are comparing the bulk tissue transcriptome on one hand and the single cell gene specificity coming from a healthy mouse. So we need to make sure that these genes remain specific to vasculature and fibroblasts even during the disease. So we looked at collagen 6, which is specific to perivascular fibroblasts. And thanks to Professor Eleonora Aronica in Amsterdam and her brain bank, we were able to see that, yes, even in ALS patients, the collagen 6 remains specific to blood vessels and becomes accumulated, as well as SPP1 protein, also known as osteopontin. It is specific to perivascular fibroblasts and becomes accumulated around blood vessels. And we were also able to see that, yes, this also happens in the mouse as early as the gene induction. When we looked at the structure of these blood vessels in the ALS patients, these were somewhat different, because here you see the collagen 6 relatively tight around the blood vessel, and here you see as if these basement membranes are separating. Right? We know that there are two basement membrane layers, and we can now stain for the specific markers for the astrocyte basement membrane layer and the mural cell basement membrane layer, like laminin alpha-1 specific for astrocytes, or collagen 4 specific to mural cells or endothelium. And we can see that in ALS, indeed, these two layers are separating. We were able to see that in these markers that we looked at, the markers of fibroblasts like collagen 6A1 and SPP1 are accumulating within these enlarged perivascular spaces. So this made us think that these enlarged perivascular spaces, and the enlargement is quantified here, are the location of these fibroblasts. And apparently the activated fibroblast secretes those proteins like SPP1 and collagen 6 inside those enlarged perivascular spaces. And this made us think, okay, so if the perivascular spaces are filled with CSF, and CSF should drain to systemic blood circulation, perhaps these proteins could be biomarkers for, well, vascular injury in ALS. But we didn't have much of patient samples available. We just had tissues from mouse and humans. So I had to reach out to a lot of clinics around the world asking for plasma samples. And here was the ALS collaboration network that I managed to put together. These groups provided us with plasma or CSF, as well as the clinical descriptions. So we had a substantial amount of plasma, but we didn't have a high throughput method to analyze it. And then I reached out to a group at the SciLife lab led by Professor Peter Nielsen, where they have a suspension array proteomics method where beads can be coupled to antibodies. Each bead has an individual fluorescent label, and you can analyze approximately 380 targets in one vial of, let's say, a few microliters of a plasma. So we had a bunch of plasma, and we had a good high throughput method for protein detection to look for the vascular injury biomarkers. And when we looked at the SPP1 levels in patients and controls, it turned out that the upper level of SPP1 can indicate shorter survival. Above this threshold, SPP1 was showing poor prognosis at the first clinical visit. And this is the combined result from clinics in Germany, Belgium, and Netherlands. We were able to confirm this in an independent labeling experiment from another cohort from patients here in Sweden, and we saw a similar effect. But you can argue this is just an arbitrary cutoff to make two arms of the Kaplan-Meier, right? High versus low. Does the effect of SPP1 increase really happen across the range of protein from the lowest to the highest? So for this, we used the proportional hazard models where incremental increase of SPP1 showed high hazard ratios for poor survival. And we compared this against these typical indications of poor survival like bulbar onset. And it turned out that this hazard ratio is pretty high, and it is higher than the neurofilaments in CSF, which are the typical traditional way of predicting survival, right? Because the moment the neurodegenerative disease happens, the neurons are dying, they're shedding the neurofilaments to CSF. And this is how we tend to predict survival at the beginning of the disease. So when we compared the hazard ratios between neurofilaments in CSF and SPP1 in plasma, it turned out that SPP1 in plasma is a pretty good survival biomarker. But you could say, well, this is a univariate model, meaning these effects are not balanced against each other, right? So maybe what the vessels are doing, that they're reflecting that the neuronal damage that has already happened. So we made a multivariate Cox model where these effects are balanced against each other, and still the increase of SPP1 in plasma, independently of levels of neurofilaments in CSF, was the highest predictor for survival with very strong hazard ratios. This could probably mean that blood vessels and their dysfunction or injury, as I like to call it, are an independent source of prediction for survival in ALS patients. So they might actually improve patients' predictions in the future. So I tend to believe that the vascular injury in ALS, it's not just a bystander effect of vessels reacting to neurons. I think they're actually happening, these changes in blood vessels are happening much earlier than we anticipated. And perivascular fibroblasts are one of the key cells responsible for this vascular injury response. When we had the group of all the transcriptomes sorted to specific cell types, we created a web resource where you can look up your mouse model or ALS patients, as well as the cell type of interest out of these 10 cell types. And then you can rank these differentiately regulated genes to the specificity and the strength of induction in ALS. Because this resource, we believe, will help other people, not necessarily interested or specializing in neurons, but those that are looking at microglia or blood vessels. They could look up if there is a vascular response and how early and which gene is responsible for these vascular changes in ALS. So looking onwards, I think that even though today ALS is still understood based on late disease events, like the neuronal phenotypes, the motor phenotypes, I think that the early vascular mechanisms in ALS could help us understand maybe not the origins of the disease, but at least the early pre-symptomatic events happening in mouse models and perhaps even patients. The problem with ALS at the diagnosis is that it's highly unpredictable. We cannot really say how long a given person will survive, particularly based on just the neurofilaments. So combining vascular response and the neurofilaments in plasma could improve the predictions of patient survival. And the moment we will have improved predictions of survival, despite the fact that ALS remains without effective treatments, having good predictions of survival will improve the evaluation of clinical trials. So I think that from now on, we should pay more attention to blood vessels in ALS and perhaps other neurodegenerative diseases than we were doing so far. So I'd like to thank everyone who was involved in this study and the few funders that were supporting us. If you have any questions about this work, you can contact me on Twitter or write me an email. Thank you. Let me just say one more thing. How did we get there? How does someone who had no resources, no group, was able to pull together a story that was, I think, valuable for the insights in ALS? Before I could tell you, there are three things that you need to know. I call them the inconvenient truths of working in academia. The first inconvenient truth, it was coined by Dr. Sidney Brenner around 1980, when he said, progress in science depends on new techniques, new discoveries, and new ideas. Probably in that order of decreasing importance. But he's saying your resources matter more than your ideas. It's interesting that he said that long before the human genome was sequenced, long before we had high throughput methods. I think what essentially he is trying to say is that your ideas don't matter as long as you have resources. Because you can always throw the latest technology on a tissue and get a seemingly meaningful result. And this gets comments like this on Twitter, right? I propose a new drinking game. Every time a paper shows single-cell RNA sequencing reveals or identifies, it means that you don't need to have a biological question in order to be productive. right? As long as you have resources. So the first rule is have resources. The second rule that we are being taught is that you should have a position with titles that allow you to recruit other people, right? I call this the leadership threshold. It is very difficult to improve your productivity just as a postdoc, right? If you want to bring your ideas to life, you're told that you're supposed to have a position that allows you to recruit people. And the third thing that we hear so often is that we should be the smartest, all-knowing person that has mastered many methods before you can even make insights in your field. So here they are, the three inconvenient truths, which we're told we cannot be productive without. But I'm here to tell you, they're not exactly truths. I think they're false. I think that, yes, you can be productive without resources, without positions with titles, and being smartest. So let me break it down for you. How can you be productive without your own resources? A few years ago, what we saw by staining ALS mice is that there's a funny phenotype happening in the basement membrane layers, right? That they're seemingly separating. And we didn't know what to do with this because this didn't react to any interventions. It was just happening. So eventually we just put it in the shelf and it never got published. And then we had access to some microarray data, which I was analyzing in Excel by copying and pasting the row of data so I could see a graph of gene expression. And I saw that collagen 1 gets activated very early, like the first month of a mouse model for ALS has collagen activated. So I thought, this could be an interesting project, but it could use some bioinformatics. So I went to Nathan Skeen and asked, can you spare some time and do a proper bioinformatics analysis of gene enrichments? And for him, it wasn't much of an effort, so he did this in a couple of days. And I thought, oh, this project is getting interesting, but we should perhaps check this genes and proteins specific to fibroblasts in patients. So I reached out to Professor Eleonora Aronica in Amsterdam, and she was happy to share a few sections of ALS patients because it wasn't much of an effort for her. And we came up with all the histology data. And then I thought, okay, but we're just looking at 4 patients. How can we make this scalable? So on conferences, I reached out to all these head clinics in ALS in all sorts of countries, and they were pretty happy to send us a few plasma samples from ALS patients. And then we had the plasma samples, but we didn't have a high throughput method to look at them. So I reached out to the SciLife Lab group of Professor Peter Nielsen, which had the bead arrays. So it is just like the stone soup. If you can make few things come together, you can actually invent your own resources. The second inconvenient truth is that you're supposed to get position with the title that allows you for recruitment. I don't think this is true. I think that even though you, at the face value, you look at the list of authors, it seems that I am a person who is hiring a lot of people, that's paying salaries of, well, at least a few of those. But in fact, I was paying only my own salary. Everything else was based on collaborations. Collaborations with KTH, collaborations with some master students that were visiting the lab, the Karolinska Hospital, other hospitals, Netherlands, Germany, and Belgium, as well as the UMIO group, which gave us the ALS mouse tissues, and some other people at KI who helped us with data analysis. So if you can't employ people, ask for collaborations. This is also kind of a lesson on how to lead without having official authority. Again, I didn't have an official authority as an employer to any of those people. So this situation really teaches you how to ask for favors, how to ask for feedback without having any official authority over any of those people. And the third inconvenient truth that we hear often is that you should be the smartest, all-knowing individual. But I don't think it works. And if you're not the all-knowing individual, you should rely on other people with diverse expertise. What you should know is you should have your core competence in a field in order to ask a relevant biological question that has a potential value. That core competence, I believe, is enough. Because individuals know relatively little, but the human collective knows quite a lot. I believe your job, if you want to run a group or be productive in science, your job is to bring the pot and drop in a stone. You need to know the core problem or the initial idea you want to solve. You need to bring meaning to the people you invite for collaboration. They need to be acutely aware of the value of the thing that you're going to do, and it should be easy enough for them to collaborate. So you need to create the mentality of abundance rather than scarcity for all people involved. And this makes the whole greater than the sum of its parts. Because if we were to publish the transcriptomics separately, the pathology separately, and the plasma profiling separately, this wouldn't make the kind of a story that it has become once we make the whole greater than the sum of its parts. So thank you for your attention, and I look forward to your questions and comments. And by the way, we are hiring. So if you're about to start a postdoc, please talk to me after. Thank you so much.